Anyone who has taken a long break from physical training knows the initial return is difficult, but they also quickly realize that regaining lost muscle size, strength, and oxidative capacity happens significantly faster than building it the first time around. For decades, sports scientists attributed this rapid reacquisition entirely to motor learning. The consensus was that the central nervous system simply remembered how to perform the movements. Recent in vivo microscopy and molecular data prove this explanation is incomplete. Skeletal muscle actually possesses its own localized, independent memory. Muscle memory operates as a tripartite biological framework. The body retains adaptations across three distinct biological domains: neural routing, cellular architecture, and epigenetic software. Examining these three tiers reveals the specific biological modifications that allow the body to preserve the results of previous training. The central nervous system acts as the baseline coordinator for every muscle contraction. Through repetition, structures like the cerebellum and the motor cortex optimize complex movements. They gradually organize chaotic neural signals into highly synchronized pulses that recruit motor units with maximum efficiency. During a prolonged period of inactivity, these optimized recruitment strategies become dormant. The neural pathways themselves, however, remain intact. When training resumes, the central nervous system rapidly reawakens these existing pathways, restoring coordination and firing rates almost immediately. This neural reawakening explains the rapid initial spike in force production. It fails to account for the accelerated physical expansion of the muscle fiber itself. Skeletal muscle fibers are a distinct anatomical structure known as a syncytium. A single fiber contains hundreds of individual nuclei, with each nucleus governing protein synthesis for a specific volume of the cell, called a myonuclear domain. To observe how a hypertrophic stimulus alters this syncytium, researchers in a 2013 study led by Ingrid Egner analyzed the muscle fibers of mice. Female mice given testosterone saw a 66% increase in myonuclei and 77% in cross-sectional area. After 3 months of withdrawal, mass shrank to baseline, but the extra myonuclei persisted. When overloaded, previously treated mice grew 31% in 6 days, compared to the control 6%. Biologists define this phenomenon as myonuclear permanence. Subsequent human resistance training studies corroborate this mechanism, demonstrating that human muscle fibers similarly induce myonuclear accretion that is maintained through long periods of detraining. During the retraining phase in humans, the local density of these retained myonuclei tightly correlates with the ultimate degree of hypertrophy achieved by individual fibers. These retained myonuclei act as permanent cellular hardware. They provide a dormant architectural framework ready to activate immediate protein synthesis the moment exercise resumes. How exactly does the muscle fiber acquire these new nuclei in the first place? The standard mechanism relies on satellite cells. Situated on the periphery of the fiber, these cells activate, proliferate, and physically fuse to the muscle tissue to donate their nuclei. This diagram illustrates a second function for satellite cells, niche orchestration. Beyond donating nuclei, the cell secretes extracellular vesicles containing microRNAs like miR-206. These vesicles migrate to neighboring fibrogenic and endothelial cells, regulating the extracellular matrix and influencing new capillary development. When satellite cells are depleted in experimental models, the muscle can temporarily adapt to exercise, but long-term adaptation ultimately fails due to fibrotic accumulation and matrix dysfunction. Satellite cells function as both architectural donors and environmental managers, ensuring that the fiber and its surrounding niche are primed for rapid expansion. While resistance training builds cellular hardware, endurance training leaves a completely different biological trace. In a clinical trial, subjects underwent 8 weeks of interval training, followed by 3 months of complete inactivity. As expected, VO2 max improved significantly during initial training, dropped entirely during the detraining phase, and rebounded with incredibly high efficiency upon retraining. The underlying molecular mechanism driving this is DNA hypomethylation, the process where tightly coiled DNA strands shed small molecular attachments, unwinding to expose genetic code. The initial 8 weeks of interval training induced this hypomethylation at 3,190 specific CPG sites across the human genome. This chart tracks specific genes, ADAM19, INPP5A, and MTHFD1L. The bottom dashed line reveals that hypomethylation was retained during 3 months of inactivity. Because methylation stays low, the corresponding solid line showing mRNA expression remains elevated, keeping metabolic and mitochondrial genes highly active. DNA hypomethylation acts as the software of muscle memory. It keeps metabolic genes transcriptionally unlocked and primed for immediate action the moment training resumes. Muscle memory relies on this exact sequence: optimized neural routing, retained cellular hardware in the form of extra myonuclei, and primed molecular software through epigenetics. It is a compounded, multi-scale biological mechanism. This biological reality creates immediate friction with real-world athletic regulations. The principle of myonuclear permanence severely challenges standard 2-year doping bans. If an athlete uses anabolic steroids even briefly, they acquire a dense myonuclear framework that persists long after the drug clears their system. This grants an architectural advantage over clean competitors that may last for a decade or more. Beyond elite sports, these mechanisms provide a direct intervention for sarcopenia, the age-related loss of muscle mass that impacts the general population. As humans age into senescence, the biological ability to generate new myonuclei from satellite cells sharply declines, largely due to reduced Notch signaling. Individuals who train rigorously in their youth build a deep structural bank of myonuclei and highly favorable epigenetic profiles before this decline begins. This structural reserve allows for much easier maintenance and recovery of muscle mass in old age, acting as a crucial buffer against physical frailty. Every period of training creates a structural and molecular reservoir, allowing the body to bypass the slow process of initial adaptation when exercise resumes.